Welcome back, class. So in this video, we are going to talk about how the cell cycle is regulated. So making sure that our cells are not dividing when they are damaged, making sure that damages can be recognized and fixed, making sure that cells grow when they need to grow, and they don't grow when they don't need to grow. Within the cell cycle of different cells, of different organisms, we can see differences in the overall length of the cell cycle. So the cell is not just constantly turning over as fast as it can. Some cells are going to take longer and some are going to be shorter. Some have different relative lengths of time. So they might spend 20% of their time in one step of the cycle. Another cell might spend 50% of its time in that step. So the different phases could be individually longer or shorter compared to other cells. And how closely mitosis and cytokinesis are coupled. Are they happening at the same time? Is cytokinesis happening afterwards? Is it a little bit earlier or a little bit later? So the cell cycle is really regulated for each cell type and each organism, that their needs will be met so that the cell is growing correctly, that the cell type is performing its job, and the organism is allowed to grow and thrive in its optimal health. At one extreme, we have cells that are constantly having to divide because they're constantly lost or constantly destroyed. This would include things like sperm formation. Men are constantly able to form sperm. Stem cells are constantly dividing. They're going to create blood cells. We constantly needing more blood cells. Skin and epithelial cells. These are constantly being worn out, shed, destroyed. So we're constantly having to create new blood, skin, and epithelial cells. At the other end, we have cells that grow very slowly, or maybe not growing at all. So mature nerve cells generally are not going to divide. Muscle cells generally are not going to divide. Then in the middle, we have cells like liver cells that if a person, donates a lobe of their liver, their cells will be stimulated to grow and their liver can regenerate itself. But a liver is not typically going to be trying to grow something when it doesn't need it to. So some cells are not going to divide unless they're stimulated. Those liver cells, lymphocytes, when they need to, they'll divide, but otherwise they don't. One of the biggest variations we see in generation time is G1. G1 can be very short. Some cells can even skip G1. G1 can be very long. Some cells can stay in G1 for, long periods of times. Here they could jump out of G1 and go into that G0 phase when they are not having any signal to replicate. They're still able to replicate if that signal shows up, but a cell could spend days, months, years, could spend almost its whole life in that G0. Although G1 is a very variable part of the generation time, the length of time in S and G2 can also be variable. Usually, cells are going to grow before they divide. Growth is typically a part of the cell cycle, but the two are not required. You can think of like a developing embryo just going through cleavage. As soon as that egg is fertilized, cleavage occurs with cell division without growth. So it does not have to be there. There's a protein called TOR. TOR stands for targeted rapamycin. We can just call it TOR. This is a protein kinase. Protein kinases add phosphate groups to molecules. And that is an important way that cells are going to signal through the cell cycle here. So this protein kinase TOR is going to be involved in the signaling that is both coordinating cell size and coordinating the cell cycle progression. TOR is going to be activated in the presence of nutrients and growth factors. So there is some signal saying you are safe to divide, it's time to divide, that activates TOR. Then TOR will allow the cell to increase its cell size and to continue through the cell cycle. So TOR, when it's activated, is able to stimulate molecules that increase protein synthesis. By increasing protein synthesis, you are increasing the mass of the cell, which is going to make the cell bigger. TOR is also able to stimulate molecules that encourage DNA replication. It's going to take the cell into the S phase. So that is how TOR is going to regulate the progression through the cell cycle. Activated TOR does both of those to allow a cell to go through a healthy cell cycle and have two healthy daughter cells created. There are several transition points along the cell cycle that are going to be important spots where we stop and check the cell before it grows. This includes this G1 going into S. That's a big one. That's called restriction point, or in yeast it's called start. Then we have the G2 to M transition. That is going to allow a cell that has already copied its DNA to go into mitosis. And then there's the Metaphase-Anaphase transition. These are three very important transition points along the cell cycle, and they're going to be very carefully controlled. These controls are going to ensure that each event of the phase are carried out in the correct order and at the correct time. So, for example, we are not going to start anaphase here unless all your chromosomes are properly attached to the spindle. We're going to make sure that all of our cells have their chromosomes lined up on their metaphase plate before they're able to separate their chromosomes and go through anaphase. And make sure that each phase is completed before the next one begins. So here we make sure that we have completed growing and copying our DNA before we go into mitosis. And we're going to make sure that we are responding to external conditions. So here this restriction point is going to be influenced by having the proper growth factors and the proper nutrients available before you commit to dividing into two cells. So that first one, the G1 to S transition, or the restriction point, is the very big key point that we're going to talk about. The conditions here determine if it will or will not proceed. In animals, this control point is called the restriction point, and it requires extracellular growth factors. That means that other cells have to send the signal that it's time to grow. The cell can't decide for itself to divide. In yeast, this is called start. Cells that are able to pass this restriction point are then committed to the S phase. They are going to copy their DNA then. If they don't pass the restriction point, they can opt out and enter into the G0. Here you can see G0 is a cycle where it can just hang out on G0 for as long as it needs until it gets those appropriate influences to trigger it to go into S. Those cells that opt into G0 are going to stay in G0 until there is a signal that allows them to go back into G1 and pass through the restriction point. Next, we have the G2 to M boundary. This is when the cell commits to entering mitosis. Some cell types have a little phase off of G2 that could be analogous to G0, where they can kind of be arrested in G2 indefinitely. But in most cells, if they're going to arrest in a non-dividing state, that'll be a G1 arrest. But G2 arrest can be seen, and it can be even more important in some cells like frog eggs. Then a third transition point is moving from metaphase to anaphase. This is when you are committed to moving your chromosomes to opposite poles to create two new cells. To be able to pass this transition, your chromosomes have to be correctly attached to the spindle. You have to have all of your chromosomes attached to those microtubules, attaching them to the spindle. Otherwise, when you go through anaphase, your chromosomes won't be properly sorted. You could end up with broken chromosomes. You could end up with chromosomes that have too many chromosomes moved to one side of the cell or not enough moved to the other side. So you have cells with inappropriate numbers of chromosomes, which can lead to diseases, especially if this is happening in like an embryonic development or in a germ cell development kind of situation. We're able to understand how the cell cycle controls work because of some different experiments that have been done. One is by experimenting on heterokaryon. So if you remember this word karyon as kind of the biology way of saying nucleus, that kernel, and hetero means different. These have two nuclei from different steps of the cell cycle. So we are going to fuse them into one cell. So here we are fusing a nucleus from the S phase with a cell whose nucleus is in the G1 phase. Here we're going to fuse a cell with a nucleus in the M phase and a cell with a nucleus in the G1 phase. So when those are fused together, you get a heterokaryon, a cell with two separate nuclei. And when you do this, if you have a cell in the S phase and a cell in the G1 phase, that G1 nucleus is going to immediately jump into the S phase. So something in this cell, some cytoplasmic signal, is telling the nucleus to divide. Even for this G1 cell, that signal is telling it, you need to go through the cell cycle, time to start S phase. If you take a cell with a nucleus in the G1 and a cell with a nucleus in the M and fuse them together, that will trigger this G1 nucleus to jump right into mitosis. It hasn't even copied its chromosomes yet. It's not ready in and of itself to go through mitosis, but there is some signal in the cytoplasm here that gets to this nucleus and tells it, you're now going to go through mitosis. So this is how we know, these heterokaryon experiments, is how we know that there are cytoplasmic signals that trigger that nucleus to go through the appropriate stage. We also can study the cell cycle in some yeast mutants. So budding yeast, their scientific name is Saccharomyces cerevisiae. There were temperature-sensitive mutants created. So I want you to think, if you made a cell that had an error in its cell cycle, it'd be kind of hard to study it because it wouldn't be able to go through the cell cycle. It wouldn't be able to grow. So they solved this problem by making these temperature-sensitive mutants. These mutants at lower temperatures, at cooler temperatures, are just fine at growing. But that mutation really kicks in at higher temperatures. It's just higher temperatures where the cell cycle is blocked. And so by using this, they can grow the cells and then study those different mutations and what they're doing. And so they were able to identify several genes that were involved in regulating the cell cycle. Paul Nurse did some similar research in a fission yeast, Schizosaccharomyces pombe, and he identified a gene called CDC2. And CDC2 is critical for that G2 to M transition. And the CDC2 molecule has counterparts in all eukaryotic cells. That is going to tell you it is very important that it is conserved across all these types of cells. And it encodes a protein kinase. Okay, that's where we're going to pick up in our next video. We're going to talk about protein kinases called cyclin-dependent kinases and the way that they are controlled with proteins called cyclins. I'll see you then. Bye.